Hi, my name is Jed Eberly. I'm an assistant professor at the MSU Central Agricultural Research Center. And I'm here today with my graduate student, Joseph Kibbiwot, to talk about some of the work that we are doing with lentil production.
My name is Joseph Kibbiwot, and I'm a graduate student at the Montana State University in the Department of Plant Science and Plant Pathology. My research is on lentil production. Montana is the leading state in lentil production in the country right now. And in 2019, there were about 320,000 acres of lentils grown in this state of Montana. Of the 320,000 acres, some of it is organic, and unfortunately, that data is not captured in any agency reports, so we don't really know the amount of organic acreage that is grown here in the state of Montana. There are several challenges that organic crop producers face, especially with the lentil crops and other pulse crops, because for one, lentils are very short plants, and though they put on some foliage, that foliage is not competitive to the weeds compared to other crops like cereal crops, including wheat and barley.
My name's Spencer Fisher. I own a company called Fisher Farm Industries, Inc. And along with my brother Logan Fisher, who owns Fisher Ag Ltd., we grow all types of different organic crops and conventional crops here in north central Montana, anywhere from south of Rudyard, 12 miles to all the way 35 miles north of Guilford. Well, lentil production is very, very finicky to start out with, and that's whether you're in organic or conventional. The weeds are ever present, especially in lentils. It seems like there is just an overabundance of Russian thistle in any field that you get into. And so with conventional, you have a little bit of time on the front end with your pre-spraying to try and take care of some of that, but you're not guaranteed it. And if they don't start out with a good stand right away, you're almost guaranteed to have weeds later on. And so just magnify that with organics, just because you don't have the opportunity to take care of it with any kind of spray. So basically all you're banking on your tillage doing a good enough job, and your seeding to do a good enough job to have a decent stand out there. If you get a jump on the weeds, they don't compete well, especially with a nice bushy stand of lentils. They're still going to be there, but they're not going to be they're not going to be such a hindrance that you can't get your crop harvested.
This field that we are looking at right now is in Rudyard, Montana, and we have other sites across the state of Montana, and those three locations are different in terms of the local environment, the rainfall amount they receive, the soil conditions in those environments are different, and all that is part of our evaluation.
Given some of these challenges facing lentil growers, we developed a research project with the following goals. The goals of this project were to 1. Evaluate the effects of different inoculant rates on nodulation, 2. Compare yields between different seeding rates, and 3. Determine the effect of seeding rates on weed density. So lentils can be inoculated by naturally occurring rhizobia in the soil. In the photo on the left, we have uninoculated lentils. Notice how there are some nodules present. This field was previously in CRP, and naturally occurring rhizobia were able to successfully nodulate the plants. In the photo on the right, you can see how much more abundant the nodules are on the inoculated plants. Notice also how much more soil is adhered to the roots with the plants that were inoculated with a mix of rhizobia and mycorrhizal fungi that are able to colonize the roots. You can see that the inoculated plants were taller and had more branching. In spite of some of the observed differences in nodule counts, our results last year showed no difference in yield or protein in response to the double inoculation. Now I would like to discuss differences in seeding rates and show some examples of how that affects yield and weed density. Lentil seeding rates can affect canopy closure and influences how effectively the crop can compete with weeds. Here we have a comparison of large green lentils at two different seeding rates. On the left is a plot planted at 100 plants per square meter, while on the right is a plot seeded at 500 plants per square meter. Notice how the denser stand provides better ground cover and can more effectively compete with weeds. However, there's a trade-off here. At lower planting densities, you tend to have more branching, while at higher planting densities, there's less branching on individual plants, as you can see from this example here.
Yeah, so what we have here, we have the black beluga lentils, and on to my right, we have a plant that came from a population density that was seeded at 700 plants per square meter, and to the left is a plant, similar variety that came, this is a black beluga lentil that came from a population that was seeded at 100 plants per square meter. The difference here is you can see this is just one plant with almost no branching, but here we have an enormous amount of branching that we can see. This shows that, when you seed at a lower seeding rate, the plant is able to compensate for that space gap. And what we want to know is, and that is part of the research, is how much can this plant compensate for the yields that it acquires just by sheer branching, as compared to when you have a lot of plants in say 1 square meter versus having few plants in 1 square meter that can compensate.
So does this increased branching compensate for the lower seeding rates compared with higher rates? Our initial results suggest that it does not, and that higher seeding rates provide a yield advantage. Our results last year showed that increasing the seeding rate from 100 to 500 plants per square meter resulted in an average yield increase of 46%. In fact, our results demonstrated a linear increase in yield in response to increasing seeding rates from 100 to 500 plants per square meter, and this was consistent across all environments and varieties. Similarly, we saw a consistent decrease in weed density from around 22 plants per square meter at the lowest seeding rate down to around 12 plants per square meter, which represents a 40% decrease in weed density across all environments and varieties. So this is the second year of the study, and in the following years we hope to expand this work to also evaluate seeding date and tillage practices. So with that, I would like to acknowledge support for this work. In addition to the MSU College of Agriculture and maize, this work was made possible by a grant from the Organic Farming Research Foundation with additional support from Timeless Seeds.